Think about the sheer volume of information you process on a regular Tuesday. You navigate traffic, read emails, have conversations, and choose what to eat for lunch. By next month, almost all of those specific details will fade into static. But if you were to win the lottery in the middle of that ordinary Tuesday, that exact sequence of events would be etched into your mind for decades. To understand how the brain distinguishes the disposable from the permanent, we have to look at the hippocampus. This seahorse-shaped structure acts as the brain's internal recording device, processing episodic memory, the ability to remember personal experiences in chronological order. While you are awake, the hippocampus is operating in its tracking mode. It diligently logs where you are, what happens, and in what sequence, continuously updating a rich cognitive map of your physical environment. The problem is, this continuous recording generates massive amounts of temporary data. You don't need a permanent record of every single footstep you took on the way to the grocery store. Since the brain simply lacks the capacity to keep an entire day's worth of raw sensory input, it requires an aggressive, highly selective sorting algorithm. It needs a way to filter out the noise and flag the exact moments that actually matter for survival or learning. The actual long-term storage facility for those important memories is the neocortex, the heavily folded outer layer of the brain. But getting data into the neocortex requires highly specific biological conditions. The cortical networks only become receptive to forming the new, permanent synaptic connections needed for long-term storage when the brain is in an offline sleep state. This creates a severe logistical bottleneck. Throughout the day, your hippocampus is rapidly acquiring new information, but the permanent storage drive in the neocortex is currently offline, remaining non-receptive to incoming signals from other brain regions. It is only hours later, when sensory input quiets down during sleep, that the neocortex enters the physiological state required to receive and integrate incoming data. This leaves us with a difficult biological puzzle. If the storage center is non-receptive while you are awake, how does the hippocampus hold on to a specific important event that happens at noon, so it can be saved at midnight? The brain has to rely on a delayed transfer pipeline. To ensure an important daytime experience survives until the neocortex becomes receptive, the hippocampus has to physically bookmark that specific sequence in its local circuitry right as it happens. To observe this bookmarking process in real time, researchers designed an experiment targeting episodic memory. They placed mice in a figure-8 maze and required them to alternate left and right turns to earn a reward. As the animals learned the route, the research team directly recorded the electrical activity of 400 individual neurons simultaneously firing inside the hippocampus. Raw electrical spikes from a single neuron tell us almost nothing. We must look at the collective population activity. With 400 recorded neurons, each cell's firing rate acts as an independent variable, mapping out to a massive 400-dimensional space. It is impossible to look at a 400-dimensional cloud of data and identify a memory. Standard graphing techniques completely fail when trying to interpret this level of simultaneous complexity. To isolate the underlying memory, researchers had to strip away the noise. They needed a way to locate the hidden mathematical shape that restricts how these neurons are allowed to behave. Fortunately, neural networks do not fire randomly. Their physical synaptic connections and the sensory inputs they receive restrict their activity, forcing the population into highly coordinated, predictable patterns. To expose these patterns, scientists applied a nonlinear dimensionality reduction technique called UMAP. UMAP searches for patterns clustering in that 400-dimensional space, collapsing the data down to 3 visible dimensions. What emerges is a structured geometric manifold mapping the brain's activity. Remarkably, this mathematical loop perfectly mirrors the physical layout of the figure-8 maze. It went even further. When organized by the animal's progress over multiple days, a clear trajectory appeared across the manifold, physically mapping out the learning process as the mouse improved at the task. This UMAP manifold functions as a reference frame. Researchers can now take any isolated burst of electrical activity from the hippocampus, project it onto this 3D structure, and identify exactly where the animal was and what sequence it was recalling. With the map established, we can finally look at exactly what happens inside the brain's circuitry the moment the animal pauses to consume a reward. At this moment of rest, a massive, synchronized wave of excitatory input sweeps across the entire hippocampal network. This is the sharp wave. Inhibitory interneurons rapidly spring into action to suppress this sudden surge of excitation. Acting as gatekeepers, they leave only a narrow window for select neurons to fire. This interaction creates a tight electrical ripple, riding the crest of the slower sharp wave. This creates a competitive environment within the network. Because inhibitory gates restrict overall activity, only the strongest, most recently reinforced synaptic pathways, the ones used to find the latest reward, survive the suppression to fire. The sharp wave ripple acts as a temporal compressor. It forces seconds of real-world behavioral navigation through a biological bottleneck, shrinking the entire experience into a dense neural sequence lasting just 100 milliseconds. So, what actual information is contained inside that compressed 100 millisecond burst of activity during a waking pause? To find out, researchers took the neural spike data generated purely during these awake ripples and began projecting it directly onto the UMAP manifold. The resulting trace perfectly matched the exact physical path the animal had just successfully navigated to reach the reward site. The entire recent memory was being fired in precise temporal order, played on extreme fast-forward through the localized circuit. These awake ripples are highly structured replays of successful actions. They act as immediate bookmarks, tagging the recent memory directly into the hippocampal circuit while the animal is still performing the task. At the end of the day, the animal falls asleep, sensory inputs quiet down, and the brain enters a dramatically different operational state. As established earlier, this offline period is exactly when the neocortex enters its receptive phase, becoming physically capable of forming new, long-term connections. During this sleep state, the researchers recorded hundreds of new sharp wave ripple bursts firing inside the hippocampus. Plotted onto the figure 8 manifold, researchers could accurately decode the sleeping brain. The new sleep traces showed striking similarity to near-wake traces, replaying the exact trial sequences and physical locations tagged earlier in the day. This geometric match suggests that the hippocampus successfully bridged the temporal gap, holding the bookmarked sequence until offline processing was possible. This provides an elegant solution for why the brain cannot simply consolidate memories the moment they happen. Transferring a pattern to the neocortex for permanent storage requires dozens of rapid, uninterrupted repetitions. A waking brain, busy constantly navigating its complex immediate surroundings, simply cannot afford to pause and endlessly loop a single memory. Instead, the awake ripple induces local synaptic plasticity. By briefly firing the sequence on fast-forward, it physically alters the local circuitry, carving a preferred path through the hippocampal network. Once asleep, the network quiets down. Without external sensory data to distract it, the circuitry naturally falls into these pre-carved paths, repeatedly triggering the exact memory and broadcasting it upward. Because the sequence is compressed to roughly 100 milliseconds, it arrives in the precise temporal windows required for the neocortex to physically rewire itself. Memory selection is a masterclass in structural dimensionality reduction, distilling vast, chaotic daily experiences into heavily compressed, localized bookmarks, optimized for delayed transfer and permanent offline storage.